Genetics has also shown us how improbable contact was, with cases like that of malaria. It has been with tropical Africans since the beginning of human time and has spread with them wherever mosquitoes could survive. Yet no evidence of its existence in the Americas before colonial times. So whomever migrated to the Americas had to have been outside the malaria zone for so many generations that no one had exposure to the mosquitoes, the malaria, and with enough time for all genetic defenses to have been bred out. Any African American with sickle cell anemia can tell you that those genetic markers do not disappear quickly. Not even over a whole millennia. Malaria is thought to have been the greatest selective pressure on the human genome in human history. It has led to populations developing genetic traits that protect from malaria and are passed on generation to generation. Multiple genetic characteristics were formed, but I will only delve into 3. The best studied influence of the malaria parasite upon the human genome is a hereditary blood disease, sickle cell anemia. The sickle cell trait causes disease, but even those only partially affected by sickle cell have substantial protection against malaria. Another well-documented set of mutations associated with malaria is that, known as thalassemias. They too have popped up in areas of malaria, sometimes where sickle cell disease did not develop, offering a secondary form of protection. They too will accompany their descendants, in their genes, who migrate to non-malaria areas. Finally, you have the Duffy-negative antigen. It was found that the majority of Afro-descent people were Duffy-negative. 68% of African Americans and 88 to 100% of Africans were Duffy-negative, including more than 90% of West Africans. This phenotype is exceedingly rare in Euro-descent people and unknown on Native Americans prior to colonial admixture. None of these markers have been found in any human remains of Native Americans carbon dated before colonialism. Malaria, along with other world diseases, exterminated more than 90% of the population in the Americas. This clearly showed a lack of any sort of genetic protection, which would indicate a lack of African, European, or Asian presences for millennia.